just a connection to evolution. Again, the error rate of DNA polymerase is pretty low, but it's not zero. So occasionally there are some mistakes made when the DNA is copying. If that becomes permanent, meaning that organism survives and passes on to the next generation, these mutations are really a source of new genetic variation for natural selection to operate on. So for example, if you possess blue eyes can be traced back to a single gene mutation. But if you look around your family or just kind of survey people, if you're out and about, you'll see that blue-eyed mutation is very common. So you can assume that mutation has some type of a benefit. Okay. There's a couple different beliefs out there about what those benefits could be for blue eyes. One is just maybe a mating advantage. So this mutation arose during the time of the Vikings. And the Vikings, they say that maybe the Viking males actually preferred women with blue eyes. And so those blue-eyed women were mated with more often, passed that blue-eyed trait down, and then the population evolved to have more blue-eyed individuals. Also, you could make a claim for, blue eyes is usually found in more people of more European ancestry. And in Europe, there would be less sunlight, and so having blue eyes actually allowed more vitamin D absorption. One of the problems that happens in replication is, remember, if we have kind of this lagging strand over here that has multiple RNA primers in there, okay, those primers will actually be removed during replication, and then DNA fills in. But you'll notice down here on the end where we have a 5', remember, DNA polymerase can add to a 5' end. And so basically what happens in eukaryotes, since we have linear rather than circular chromosomes like a prokaryote, is with each successive replication, the DNA actually gets shorter and shorter and shorter. And so there's kind of some good news and some bad news. On the ends of your chromosomes, we do have these highly repetitive sequences of DNA called telomeres. And telomeres basically are kind of like caps on the ends of the chromosomes. So not something that you would find in a prokaryote, because remember, prokaryotes have circular chromosomes. But basically think of these caps, they don't contain any genetic information. They're just a protective end. So as that chromosome actually gets shorter and shorter and shorter, at least it's not clipping off any genetic information, for proteins that need to be made. And we do correlate these with aging. It's kind of a question mark there, but we do see the older individuals do have shorter telomeres. We also have correlated it now with stress. So they've compared the length of telomeres in children who have maybe grown up in an abusive environment, or maybe like orphanages in some countries, and they see that their telomeres are actually kind of clipped off at a faster rate. Okay. So here's kind of the good news, bad news. There is an enzyme that can come in and lengthen the telomeres and basically recap them. And that enzyme has a real creative name. It's just called telomerase. We have a copy of that gene in every cell of our body, but it's usually only active in a few cell types. So one that's missing here is, for example, stem cells of your bone marrow. So that gene is activated in there, and so that's why those stem cells can create new blood cells that have the full set of chromosomes. They're also active in germline cells. So the good news is that these are the ones that are going to give rise to your gametes and eventually your future offspring. And so, of course, we want our children to start life with a full set of their chromosomes and a full telomere. So that's kind of the good news, right? These are good applications and good uses of that enzyme. But we also find that in cancer, this gene is also active. And so that means even though a cancer cell divides more often and probably has a lot of defects in that DNA, that enzyme will come in and recap it. And so this is really what makes cancer immortal. So think about those HeLa cells, Henrietta Lacks' cells, and how they've really been dividing since 1951. Most cells reach what's called the Hayflick limit. And so it might divide maybe 50 times. And after that 50 times, the telomeres have reached kind of a critical shortened length. And that signifies to the cell that cell should basically self-destruct. And we call that apoptosis, where that cell kind of self-destructs. And it's kind of our body's way of kind of purging some of those cells that have divided a lot and maybe divided and acquired some changes. Here's a nice little organizer for you to compare prokaryotes and eukaryotes and their chromosomes. So I'll let you complete that. And then this last little section just talks about how kind of the dilemma we have in our cells. We have 6 feet of DNA. And so if you think about that, we have 6 feet of DNA that has to be compacted and folded into nearly every cell of our body. And so here you'll recognize the DNA. And the very first level of folding that happens is we have these little histone proteins right here. Okay, there are 8 of those histones that will actually come together. You can kind of see that there's 8 in here. And notice the DNA kind of winds around it about 2 times. And that structure is called a nucleosome. Okay. There's a little piece of DNA, we call that linker DNA, that's found in between. But notice how these all kind of compact right here. And so the DNA in blue is still accessible at this point. So if a gene is being actively read and products are made from it, it's probably going to be in basically this level of the folding. So picking up from that, we call this the 30 nanometer fiber. Okay, this is what you know as chromatin. It's found during interphase. Okay, but then this structure basically starts to loop. Okay, so again, we're trying to compact this. And at this point here, you really can't see any DNA. The DNA is not accessible here, so the genes here would be turned off. And eventually that even folds into this replicated chromosome that we see. In fact, this one is a metaphase chromosome. So imagine, spindle attaching to that centromere there. And so here's a little animation showing this folding.
In this animation, we'll see the remarkable way our DNA is tightly packed up to fit into the nucleus of every cell. The process starts with assembly of a nucleosome, which is formed when 8 separate histone protein subunits attach to the DNA molecule. The combined tight loop of DNA and protein is the nucleosome. Multiple nucleosomes are coiled together, and these then stack on top of each other. The end result is a fiber of packed nucleosomes known as chromatin. This fiber, which at this point is condensed to a thickness of 30 nanometers, is then looped and further packaged using other proteins which are not shown here. This remarkable multiple folding allows 6 feet of DNA to fit into the nucleus of each cell in our body, an object so small that 10,000 nuclei could fit on the tip of a needle. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope, chromosomes. It is important to realize that chromosomes are not always present. They form only when cells are dividing. At other times, as we can see here at the end of cell division, our DNA becomes less highly organized.
And then when we talk about chromatin in the past, okay, I'm talking about U or true chromatin. Okay, again, this is what we see during interphase. It's not, it's a little bit more loose. Okay, there are some regions though during interphase that are really highly packed. So when it talks about highly condensed, they're really packed with a lot of these proteins, and we call this heterochromatin. So this is where we, for example, the centromeres and telomeres, but also genes that have been shut off. So a lot of the genes that maybe formed you as a fetus, like genes that code for, how the heart was to develop, those are off. Okay, and so those would be found really tightly packed and inaccessible, and that's heterochromatin. So the form of chromatin that really makes the DNA accessible is really that loose stuff that we call the euchromatin, or just chromatin for short.